Okay, this is biochem video number 4, all about proteins. So I have this giant list because proteins are the most diverse of the organic macromolecules that we'll be studying, and they're structurally and functionally diverse. Structurally diverse means that you will notice many different forms. Each one looks a little bit different, but we'll talk about what the key identifying feature is, so you'll notice right away, ooh, that's a protein. Functionally diverse means they do a lot of different jobs. So I want you to write down this list of jobs that they do in your body. They function as enzymes in structure of your body, carriers and transporters within your blood, cell communication, signals, receptors, defense, movement, and storage. A lot of different functions. All right, structure. I know that many of you could tell me right away the structure or the building block of a protein is an amino acid. So the monomer, the single unit, is an amino acid, and there are 20 different types. A polymer means that we can make a long repeated chain of amino acids, and that is a polypeptide. And when a polypeptide forms and takes on a unique shape, then it is considered a protein. So if we start up here, this is one amino acid. Hook it with another amino acid. I know you can tell me what that's called. Here's water coming out from it. Dehydration synthesis, and that builds a dipeptide. We could keep going until we have a polypeptide, a long chain. That long chain will fold or orient itself into a very specific shape, like hemoglobin here has a unique shape. All right, amino acids. So let's talk about the building blocks. I want you to draw an amino acid. There is an amino group on one end. That is in blue. There is a carboxyl group on the other end, so two functional groups that we've talked about. There is a side chain coming off of the central carbon. There's 20 different side chains making up 20 different amino acids, and there's always a hydrogen attached to the carbon. The R group is variable, different for each amino acid. That is what's really going to confer its unique properties to the amino acid. Here are some examples. You do not need to write this all down, but you do need to know that form determines function. So if the form is that it has something nonpolar, like a hydrogen, or a carbon with 3 hydrogens, or many carbons with just hydrogens, those are very nonpolar bonds. It makes it a nonpolar molecule, and it will behave hydrophobically because there is no charges, therefore no attraction to water. So these amino acids tend to be very nonpolar and therefore very hydrophobic because of their nonpolarity. Here are some polar ones. So what do we expect to see with a polar? Charges. So here there's an OH. The OH confers a negative charge. This OH confers a negative, H confers a negative, another OH, another O. You will notice that there's something with a charge. It might not have a charge now, but it could drop the hydrogen and therefore have a negative charge. So these will be very hydrophilic. They will like water. They will form hydrogen bonds with water because of their charges. Here are some other ones. These are all hydrogen donors, which means they will act as an acid. They will increase the acidity of the solution they are in by donating an H+ ion. So this one used to have an H on it, used to have a carboxyl group, dropped the H, so truly acting as an organic acid. It increased the it decreased the pH number by increasing the H+ concentration. So each of these did this. They each donated an H to the solution. These ones are bases. They are not a base because they donate OH, but instead they're a base because they accept H+ out of a solution. So pH is constantly measuring the amount of H+ in a solution. If you remove some of the H+, you're making it less acidic. Therefore, you are behaving as a base. This one, it's typically ones with nitrogen. Nitrogen holds 2 hydrogens normally, but will take on another hydrogen. When it takes on that third hydrogen, it is acting as a base. If it has sulfur in its side chain, it is going to be important later on. We're going to talk about disulfide bridges, so two sulfurs coming together to form a connection. There are two amino acids that have sulfur in them, and they will have unique properties because of that sulfur. All right, getting into proteins. How do you build them? We're going to build them with a peptide bond. So if I just create a new screen here, because I like to draw it with you as you draw. To draw an amino acid, I need an amino group, a central carbon that has a side chain and an H, and a last carbon that has a carboxyl group. My next amino acid in line will have that exact same structure, H, H, C, side chain, hydrogen, carboxyl group. Dehydration synthesis means I am going to take an OH off one and an H off the other. I now have a spot for a bond. So the C, it's going to go N, C, C, N, C, C. And I can attach all the other things. This carbon is still going to have a double bonded O. This is still going to have an H. This is still going to have a side chain. This is still going to have its two H's. This nitrogen only has one H now. This has an H and an R. This has a double bonded O and an OH. All right, my one unique new bond is right here. That is called a peptide bond. So we've talked about different linkages when you have dehydration synthesis. In sugars, we had a glycosidic linkage, that oxygen connecting two sugar rings. In lipids, we had an ester linkage connecting glycerol to a fatty acid. In proteins, we have a peptide bond. The bond is between the carbon of the carboxyl group and the nitrogen of the amino group of the next amino acid. When you build a protein, it just goes in one direction, like one long chain. It doesn't ever come off the R's. It doesn't come off the H's. It keeps going NCC, NCC, NCC for as long as that protein is. There is an amino end to my protein. There is a carboxyl end to my protein. Again, the difference between the words polypeptide and protein, polypeptide is if you just look at the unique chain that goes amino acid, amino acid, amino acid, all connected by peptide bonds. If you use the word protein, you're looking at the final product. It doesn't stay a long chain, it folds. And when it folds, it takes on a unique shape, and I probably just have to remind you of enzymes to think of a unique shape. So an enzyme might have a shape like this, and this area here is its active site where a substrate could come in. That unique shape is because of its folding. All right, we're going to talk about their shape. Protein structure can be broken into 4 parts of structure. We call it primary, secondary, tertiary, and quaternary structure. The function of a protein is obviously so important if you remember back to that long list of jobs that they can do. So there must be many different shapes that a protein can take on. The primary structure, the first level of structure that you always need to look at is what is the order of the amino acids in the polypeptide. So what is the amino acid sequence? How do we get an amino acid sequence? Your DNA. A's, T's, C's, and G's come together as codons, 3 at a time, in messenger RNA to code for a specific amino acid. So for example, if the mRNA has a codon of CCC, that will code for just one amino acid to come to the ribosome. Another 3-letter codon like GGU will code for a different amino acid to come into that place. The order of the amino acids in a row, that's its primary structure. If you have a change in primary structure, you've got a mutation. That mutation could cause a drastic effect in your body. So, here is an example. This is how sickle cell anemia, which actually, when you're building that protein, there's 146 amino acids in the chain. This is an error with just one. The 6th amino acid that it should code for is GLU. If you have an error that causes sickle cell anemia, it causes valine to come into that space instead. This is a hydrophilic amino acid. This is a hydrophobic amino acid. If it's hydrophobic, it's going to bend in away from water and change the overall shape. And remember, sickle cells, red blood cells, have this funny shape, and the shape change is due to a change in amino acid. So the primary structure has altered. All right, secondary structure is the initial folding. Folding along short sections of a polypeptide and its interactions between their R groups, their side chains. So hydrogen bonds allow R chains to be attracted to each other or maybe stay away from each other. And it tends to take on two things. This right here is an alpha helix. I'm going to show you in class with a pipe cleaner an easy way of remembering these two. This one is a beta pleated sheet. So they are two different folding patterns that are seen in almost every protein that exists. It usually takes on one form or the other. Tertiary. Tertiary structure is how does it overall glob and make this unique form and unique shape. The whole molecule is folding over, and there are at least 3 different things that we know of that cause this unique folding. Hydrophobic, hydrophilic interactions. Part of the protein will be like, oh, I like water, and another part over there, oh, I like water. Let's bend this way towards the water. As it bends, the part that does not like water bends in. So if you picture like a hair clip. A hair clip is like this, bent over. If you straightened it out, that could be my polypeptide. And the fact that the two ends, each were hydrophilic and faced outward, and then the inner part where the bend is, we could think of that as hydrophobic. So it takes on this overall bending and folding due to hydrophobic, hydrophilic interactions of the side chains. Hydrogen bonds and ionic bonds are other ways where pluses and negatives, oh, you're a plus, oh, I'm a minus, let's get together. It'll cause it to bend and fold in unique ways. Disulfide bridges. There were 2 amino acids that had sulfur in them. If they are anywhere near each other, they will bend towards each other and very much stabilize the protein by forming a disulfide bridge. This is an example. This is a picture. This is my polypeptide chain. Each of these are representing different R groups that come off of the amino acids in the chain. Here's a hydrogen bond forming. That's going to attract the molecules to each other. Here's a hydrophobic interaction. Like dissolves like. They both do not like water, so they're going to bend in towards the middle, and the water would be out here where the hydrogen bonds are. This has got pluses and minuses, so more attracted to it. There's 2 amino acids that each had a side chain with sulfur, so it formed a disulfide bridge. And here's an ionic bond. The attraction of this O- and this NH3+, a plus-minus interaction, is an ionic interaction. Quaternary, the last level of structure, is all about one polypeptide joining with another polypeptide and maybe even a third polypeptide or even a fourth to give an overall large protein its shape. A perfect example is hemoglobin. Hemoglobin has 4 chains that make up one molecule. So the one protein hemoglobin actually has 4 polypeptide chains in it. Here are the 4 chains. There are 2 alpha chains in pink. There are 2 beta chains in brown. Each chain has little iron molecules at the center of it. Its unique shape is due to the interactions of these 4 chains coming together. That is quaternary structure, and it only is true for a molecule that has more than one polypeptide. If your protein is a single polypeptide, it will not have quaternary structure. I think that brings us to the end. Oh, here's a little review. So my amino acid sequence is my primary structure. The R group hydrogen bonding that happens allows it to either take on an alpha helix or a beta pleated sheath. That's secondary structure. How it overall folds in funky ways, that is its 3rd level of structure due to those R groups. And then the 4th is if you put together multiple polypeptides, it will take on a new form, a new shape. My last slide here, I'm sure you remember this word denaturing. If a protein denatures, it has heated up and its shape has changed because hydrogen bonds have broken, maybe ionic attractions have broken, and when they break and change, you usually cannot get your original substance back. What denatures things? Temperature, high temperature. Low just slows it down, it doesn't denature. pH, high pH, low pH can both denature. Salinity, adding salt water, increasing salt concentration alters a protein and makes it not function anymore. What's changing when you denature? The shape. It's all about the shape changing, and the shape is what allowed it to do its job. So if you alter the shape, you're destroying its function. All of these came from chapter 4 and chapter 5 in your book. So look through it, look for the specific diagrams, look to add things to your Cornell notes, and ask me questions in class.